welcome to the first session today. Like all fibroblasts or epithelial cells, every fibrosis colloquium is different. And the idea to do this is the first time, for those of you that have been to other meetings, the first time we've done something quite like this, where we've given a lot of emphasis to the posters and then had a dedicated discussion session. So this is a bit of a trial. We hope it goes well, and of course that will depend on the input from you. So there's a few technical challenges. We don't have a lot of microphones, but we have two. There's one there. Is there another one mobile as well? There's one in there. Okay. Sounds useful. So put up your hand, and we'll try and get the microphone to you while the other questions going on. Otherwise, won't get a chance to get things going. So there'll also be questions from the other room, and I've got a screen here that I can see that happening. So the key thing is to get discussions. There are a lot of you that we can't keep quiet, and we want everyone to talk, but I really want the poster presenters to present as much as possible. So I'm going to sort of look for poster presenters for the first two contributors after our moderators get things going. So it'll be free for all, but let's start with the actual poster presenters. So I know who you are. Please put your hand up and we'll get things going. So Hal Collard is going to kick things off, I think on the clinical stuff, Hal, and thanks for getting it organized for us.
Yeah, sure. Yeah, thank you, Marvin. Thank you. Okay, so yeah, as Jeff said, I think this is a new thing, and hopefully will be great. Could be a disaster. I don't think so, because I think really the goal is just to have a discussion. So what the four kind of thematic organizers talked about doing was coming up with some general themes to discuss that the posters talked about and represented, rather than trying to go through really specifics of each poster. Number one, because there's no way to do that in the time we have, but also because I think this actually is an opportunity with everyone in the room to really talk about where we are and where we maybe should be going around some important themes in fibrosis. So I tried to actually just outline a few general questions and ideas, and I'm hoping that what we can do is have a discussion amongst the group here on how we feel about that, where we need to go in the future. So I'm going to actually quote David Schwartz from yesterday. Where are you, David? Where is he? He's in the other room. He's out having coffee. So David came up at the end and said he wanted to ask a question, which was about how to deal with disease heterogeneity and the impact of disease heterogeneity on drug discovery and clinical trial design. And I actually think that sentence sums up the themes that I want to talk about or want us to talk about related to the clinical posters. So I really divide that into two things. One, this issue of disease heterogeneity or phenotyping. I think it's a very important topic. It's something a lot of posters look at. It's very related to biomarkers. And I'm hoping we can have a discussion around that part. And then talk about how that influences drug discovery, how we should approach drug discovery, and trial design, so issues of patient enrichment and so on. So maybe we'll start. Okay, good. It's doing okay? Okay. Maybe we'll start with the disease heterogeneity issue. So what do we know about this? I think the posters inform some of that. What do we know about disease heterogeneity? We think it's there, but what do we actually know? What do we need to know, and how do we get there? I think that's the place I'd like to start. And if there are no volunteers, I'm going to call names.
Let's get some poster presenters to speak to that first.
Oh, and Jeff, just to remind the people in the other room, if you have a question or comment, please stand up and stand by the microphone. We can see you on the monitor here.
So we have a brave poster presenter who's going to kick us off. Thank you.
Yeah, Antje Bosse from Freiburg, Germany. So I think one very important phenotype in IPF is whether a patient will evolve acute exacerbation or not, and whether a patient will develop acute exacerbation or not. And certainly, at least in our hands, there are biomarkers which somehow reflect the risk, or whether a patient is prone to acute exacerbation or not, and others do that not. They cannot detect the risk for acute exacerbation. So I would suggest that acute exacerbation is one phenotype we should look at.
Yeah, so to build on that, I think the acute exacerbation is the obvious and extreme example of the heterogeneity that we've observed for years, which is that some people live forever and come to our clinic month after month after month, and some die very quickly. But the key is, how do we predict that at the very beginning? So there's a lot of people who are doing biomarker studies, and CCL18, MMP7 may help, but is that enough? And then how are we going to impact on that with a therapy? So we have to have a marker which responds to a therapy. So the challenge is going to be to identify the biomarker and the therapy at the same time, and then do the appropriate study. And I think people are working on that, but it requires patient numbers and coordination amongst groups. But that's the way forward, I think.
Let me make just a comment about that before, which is this issue of phenotype. So are you saying you think the acute exacerbation, patients who have acute exacerbation actually have a different disease or a different mechanism in play, or is it just a different behavior that's not necessarily related to the kind of underlying fundamental biology of the disease? Angie, you want to just speak to that? Can you just speak?
No, I think that I think at least looking at our cohort, that patients with acute exacerbation, that this is a different disease, and I believe that there are different mechanisms involved in at least in triggering acute exacerbation, or the mechanism involved which make these patients prone to acute exacerbations might be different from other mechanism involved in remodeling or.
Okay. Yeah.
And I think that this difference, or that these two phenotypes should be reflected by biomarkers. And as I said, I think there are certain distinct biomarkers that in our hands might be able to predict it, and others might be able to predict remodeling or whatsoever.
Hi, I'm Seth Porter from Fibrogen. We have a poster there on FG3019, an antibody to connective tissue growth factor. And we've got some encouraging data in a phase 2A trial where we've seen some signs of improvement in terms of HRCT, and we've seen some signs of improvement in FVC. We're not particularly interested in a biomarker for disease. What we're interested in is a biomarker that would be predictive of response to our therapeutic. And we're taking a fairly agnostic approach to that. So we've taken a relatively small number of samples from patients who appear to be not responding, or some patients who appear to be getting worse, or some patients who are getting better, and we've subjected those to a rules-based medicine approach for looking at about 250 different markers. And so far, so it's not an exhaustive study, a relatively small number of samples. We haven't seen anything particularly jumping out at us, and we're looking for individual markers as well as looking for patterns of markers. But that's something we intend to follow up on. We're hoping to find a marker, or fingerprint, if you will, because that would certainly help us in clinical development. But we haven't seen anything yet. Which compound? FG3019, the antibody to connective tissue growth factor.
Got one down here. Hal.
Joe has, Marvin.
Yeah, just to make one point, the acute exacerbation, by the way, acute and exacerbation mean the same thing, but that's okay. So the exacerbation of pulmonary fibrosis is a clinical syndrome that we all recognize, thanks to you. And, but there are all degrees of acute exacerbation. Some of them are very subclinical. We just went through close to 700 open tissue biopsies, open lung biopsies, or explanted lung examinations in both familial and what we call sporadic pulmonary fibrosis for the purpose of seeing if there were any really differences. But one thing that was very, very striking when you do this is that it is very common to see organizing diffuse alveolar damage in the lungs of these patients, particularly the ones that are undergoing transplant. When you really look at them carefully and you have a lot of tissue to look at, there's a lot of these small areas of fibrin formation and organization. So it's a very common lesion. So I don't know if the phenotype of the very acute one, which occurs, what do we say, 10% of people or something like that, as far as we know, is really a separate phenotype or not. I just don't know.
Okay. Hal?
Yeah,
Too much.
Talmadge.
I just wanted to follow up and ask a question and make a comment. I think we need to define what we mean by heterogeneity, because I don't think we're all talking about the same thing. And so I think we should have a discussion about what is that, maybe not at this forum, but somewhere along the line, to define what we're talking about.
Do you want to take a shot at it?
No. I mean, I have my own thoughts, but I really do think we're not talking about the same thing. And the other point I wanted to make was that I worry that a lot of what we understand about the heterogeneity, the clinical heterogeneity, may in fact be wrong, because what we learned, what we understood about it actually probably had a lot to do with what we were doing to the patients, especially in the way of managing them with treatment with steroids and other drugs. And I think that what has happened, if you look at the clinical trials we've done in the last 5 years, for example, the rate of death has declined dramatically. You can say it's because of we're picking milder patients, but I think overall the rate of death has declined in this disease. And 2 years from now, we'll look back and say, during this period, something happened. What happened is we stopped poisoning the patients with the treatments that we were giving them. And the second point is you also look at the rate of acute exacerbation. It has declined. So the numbers we talk about are not the same numbers that were there 10 years ago. So I think we need to define heterogeneity, and we need to realize that we are looking at a different population of patients than what we were looking at 10 years ago. So things have changed.
So let me just use, since we don't have a lot of time and I want to make sure we keep the, let me just expand on Talmadge's and other people's points. So ask a question. Do we know anything about disease heterogeneity in fibrosis besides that it exists? I mean, can we actually agree on a definition of and examples of disease heterogeneity that we know exist, or do we actually need to take a step back and think about how we would start to address that and answer those questions? I know, actually, Dean, Joe has a question in the back. Joe, do you want to? Do you have a microphone for?
This one? Okay, sorry. Joe Aaron from Genentech. To get to your point about what do we mean by heterogeneity, I'm not sure what the answer is in IPF, but I can tell you what the answer is from our experience in asthma and my colleagues' experience in rheumatoid arthritis, which we're working on, is that you can define heterogeneity on a number of different levels, which are all sort of vaguely overlapping, but it's not cut and dried in that way. So you can talk about clinical heterogeneity. What is the disease trajectory of a patient with IPF? You can talk about biological or pathological heterogeneity. You know, what type of do they have predominantly UIP? Do they have UIP and NSIP mixed? Do they have reactive inflammation? You know, those are different things you can find when you look in the lung. But when we're talking about transitioning into developing drugs, we also need to talk about pathway heterogeneity. So if you are going to target a particular pathway, whatever it is, that pathway is more or less expressed across the population of patients. And I think we need to figure out how to link those three things. It's really hard. I don't know how to do it yet. But I don't think we can separate those three things if we're trying to really understand the disease as a whole.
So Joe, before you sit down, let me just, since I wanted to tie this to drug discovery and clinical trials, so if we take the approach of thinking about pathway heterogeneity, then can you give us some insight or thoughts, and can we as a group come up with, again, some agreement around different pathways that are important, or are we not yet there?
I don't think we're there. I think we need to do the clinical experiments, but as many people have brought up before, you could probably make the argument that almost all of the clinical trials that have been conducted heretofore that have been negative could be negative because the target was chosen inappropriately, but they could just as well be negative because the trial was not designed to be informative as to whether hitting that target was meaningful in IPF in general or in a subset of IPF patients. And so I think somebody made the point yesterday, maybe we need to go back and look at some of these failed agents and say, well, maybe there's 5% of the patients that would have benefited from this. Is there a way that we can get to that? So it's really hard, but I think to the point of drug development, we're trying to pick specific agents that we think will target something that's active in the disease process. So if you look at a biopsy from IPF and your objective is to, I'm not saying this is necessarily the right objective, but your objective is to prevent the progression of fibrosis, I think you want to target something that seems to be at least spatially and temporally located to this front of fibrosis that underlies relatively normal appearing lung. And so we actually see that that's the tack that a lot of people are taking. TGF-beta is expressed there, LOXL2 is expressed there, CTGF is expressed there. Sorry. But those targets do seem to be in that area. But then you have to assess your clinical trial in terms of, well, did you actually affect the progression of fibrosis? And I don't know that measuring FVC or DLCO or 6-minute walk test actually answers that biological question about the intervention.
Yeah, Yingzi, I'm from Pittsburgh. So you know we work on the biomarker stuff, but I think the phenotyping we definitely need to emphasize the molecular phenotyping. This is including the peripheral biomarkers, but also including the genetics. Mm-hmm. So the combine in collaboration with Denver group, my poster. Some of you came up to my poster. We did the mortality analysis and on the MARC5B. You all know the MARC5B. We published. David Group published this last year in New England Journal of Medicine. We validated, also published on the same issue, and predict the predisposition to the IPF, development of IPF. But also they are providing the advantage in mortality. So in other words, so we can also use the genetics to phenotyping. So now we have people who has the polymorphism, who don't have the polymorphism, clearly has different outcomes. So we are grouping people separately. And on top of that, you all know our paper last year in the Blue Journal about the protein biomarkers, in particular MMP7. So we derived a personal clinical molecular index for mortality. And then, if we combine the MARC5B genotype can classify people with a high risk and low risk for mortality. But then you add MMP7 into it, what happens is only the people who has a lower risk group, now you further dividing them into a higher and lower group. So I think this molecular, phenotyping and the genetic phenotyping is very important. In particular, the genetics is it's more useful because you don't have to, measure them, different times longitudinally or at different times because the genetics doesn't change. So if you do have them, you can, know them in a higher risk, lower risk, and then that will be providing a very important information. So like yesterday, the talk, there are other groups having other polymorphism, so if this is all validated, so we could have a group of, genetics markers, and then in combination with a molecular, plasma, prefer protein biomarkers will be very important.
Okay, these are great comments. My job is to police the time a little bit. I'd like Hal to sort of wrap up, maybe finish with a final comment from him, and then if there's any final burning statements, then we can finish this part of the session.
So my wrap-up question is then about the future. So I think we've talked about kind of pathway, molecular phenotyping, that it's important, that we don't really know where we're at with that yet, but there are different ideas. So I guess the question is, what do we need to do in the next 5 years? Is it to just test specific potential examples of that phenotype? I mean, or is there some more comprehensive way we can try to get at this issue that then can direct us to which pathways to look at? If there are any brief comments about that, it'd be great. If there aren't any, maybe you can all just think about it. Dean.
Okay, I'll make a brief comment. I think basically where we're at right now is we don't know how to stratify patients in advance for clinical trials because we don't have enough information about that. So I would argue strongly that what we need to do is be sure that every clinical trial has the capacity to collect absolutely as much information as possible, molecular phenotyping, genetics, and ideally in vivo imaging techniques, so that after we do those clinical trials, we'll at least be in a position to go back and figure out if there are clearly defined subgroups that actually benefited, to try to get at Marvin's point. I don't think we can do it in advance because we don't have enough information, but if we're at least collecting that information, and as a community, we're vigilant to be sure that we keep the pharmaceutical companies honest about looking back and picking subgroups that we could then design new studies for. We might be able to 5 years from now resuscitate drugs that otherwise we would have thrown out.
And Dean, do they need to be interventional studies? I mean, or would that same process be served by just following patients?
Well, the kind of longitudinal studies that people were talking about yesterday are clearly important for trying to evaluate the measurements that you're going to make. But I think the only way we're really going to know what matters is by interventional studies with new therapies.
Look, we're going to spend a lot more time on this subject over the next years, and tomorrow, actually, Ron Dubois and Martin Cobb are chairing a session. I suspect they'll come back to this theme as well. Hal, thank you for stimulating things. Sorry that we can't go on with the discussion, but, people like Talmage and many others, many of us have been in this area for a while. I think the pace of change has been a bit uneven. We see stepwise changes, and then things go quiet for a while. And, we just need a lot more data on these phenotypes. The importance has been recognized for a long time, but we just need to find ways of classifying patients that are better, and it'll come with time, but we're clearly going to have to be a bit patient. Thank you for your comments, everybody on that subject. I'm going to hand over to Gisli Jenkins, who will talk about some of the translational posters. And just remind you, we want the poster presenters to speak first. Gisli.
So my brief was to look at the translational posters. And I think that was a difficult one to classify because everyone has a different understanding of what translational is. But what came across to me was that most of the posters which were grouped as translational were using human tissue in one way or the other. So they were either looking at biopsy samples or mRNA or blood or occasionally lavage. And so the theme that I was going to try and explore in the next half hour is, how do we best utilize tissue, and is it really practical in IPF given the difficulties in collecting tissue? So I was going to start off with a sort of a simple question about biopsies, really. And so when you do a biopsy, and the immunophenotyping of that biopsy is clearly something that people regard as potentially valuable, But what does a raised marker actually mean? What does it tell us about the disease? And is it a normal response that is failing, so it's inadequate, so the protein is being upregulated in a response that's failing, and should we actually be augmenting that response? Or is it an abnormal response that really should be inhibited? And the trouble with biopsies is that it's a binary response, so I'd be interested in people's opinions on what that means.
Okay, let's get some poster presenters. So Gisli, while people are thinking about that, I just want to kick it off. So I think what you're getting at is, well, firstly, biopsies, depending on where you get the biopsy from, you're going to get different answers. So there's an issue there. And what you're getting at is as whether we're looking at sort of anti-fibrotic pathways, fibrotic pathways, and the complexity of that issue.
A lot of people, my own group included, are measuring stuff on biopsies, and it goes up, or it doesn't. And if it goes up, does that mean that that molecule is critical in the pathogenesis of fibrosis, or does it represent a generic change in the cell that is expressing that protein, and we shouldn't be trying to suppress it?
Okay. Richard Marshall, GSK.
Hi there. I didn't present the poster, but one of our group did, Carmel Nanthakumar, and just to sort of extend that, I think it's a key question. Firstly, it relates a little bit to the discussion we had before, which is what material do you have, what tools do you have to phenotype? And actually, of course, the tissue is the ultimate place to go for that. So it's a critical question about getting tissue and how we use it. And then I think to get at your question, Gisli, obviously functional experiments are one way to understand the importance of changes in expression. And I guess it touches on what Dean said, that ultimately you need an intervention to really prove the piece. So the poster that Carmel presented was, and it's something that I know a number of groups have tried and are still trying to do, is trying to understand what value you can get from functional pharmacology experiments in vitro using human tissue. And I think the answer is it will have clear limitations, many logistical, some practical, but there are some key things that could make life a lot easier for doing those studies. One is getting the tissue, and I think the other would be, I guess, cross-learnings across different groups. But that feels like one way in which you could provide an intermediate translational step. But it won't be the whole answer.
Great. Yeah, Oliver Eickelberg from Munich.
Just to continue what Richard said, if we look to COPD, and we learn a little bit from what COPD has done over the last couple of years. So remember, we in IPF have a gene signature in the lung that's been there forever. It's very strong, it's very reproducible. Naftali's initial MMP7 was done in, 6 IPF patients. It's still one of the strongest genes. Yet we clearly lack a correlation of gene expression to certain areas of the lung. We don't know whether that comes from honeycombing, fibrotic area, whatsoever. So COPD, Avi Spira's group has just published in Genome Medicine the first gene or comprehensive gene array looking at multiple databases and correlated that to quantitative histology.
Hold it close to your mouth.
Yo, better? So they've done the correlation of gene expression pattern with quantitative histology, i.e. the degree of emphysema correlating to gene expression patterns. And at least to my knowledge, this is the first clear-cut gene expression signature they have. They looked at it, they intervened therapeutically, and they found a drug that actually reversed that phenotype, at least in cells. We have not been able to do so in IPF yet, and I think that's where we need to move. We need to actually correlate gene expression to definitive quantitative histology such that we know in what areas our gene expressed in what manner. I think that's actually one of the tasks we're left with for the future.
I'm Gerd Irminger from Geneva. I'm actually rather a cancer scientist than a fibrosis person. And I even don't have a poster. But just
Could you speak up a little bit? Sorry, it's...
So I don't have a poster, but when you are talking now about biomarkers and following the disease progression by biomarkers from biopsies, I wonder, couldn't you, like in cancer biology, we are looking for markers that you can find in the blood, where you can follow up much more frequently, and that could be a diagnostic marker, but a follow-up marker and a response to treatment marker all at the same time. And I haven't seen any poster where people looked as signatures in the blood.
Okay, good point. I mean, we could have a bit of further discussion on that issue. Yes?
Andreas from Gießen. I would like to follow up with Richard's commentary. I think we are yet not at the point where we can use human samples in an appropriate way. We still have a way to go until we can look at thin sections, isolated cell types of IPF lungs. And I think this is really pivotal for us to pre-check candidates after having done some kind of preclinical animal model of which we all believe is not possible right now because we don't have the appropriate model. So I think for the future, before going to early phase 1-2 trials in humans, we should really try to set up reliable assays employing IPF materials, IPF cells, IPF thin sections, in order to study in a short term the impact of our candidate molecules on these this specimen.
Okay, thank you. It's a question towards the back there, I think. Okay, same gentleman. Keep it brief. And can you speak very loudly?
Yeah,
There's
so
problem hearing.
I mean, to the cancer biologist question, our poster actually did all of those things that you were asking about. We looked at gene expression, and we were able to localize particular patterns of gene expression to particular features. It's still a bit crude at this point. And then we've looked at peripheral blood biomarkers that correspond to those features. So it's exactly, I think, well, personally, I think that's what we need to be doing. Because when you're looking at a single biopsy, you may have sampling artifacts. If you're looking at a peripheral biomarker, that should integrate it across both lungs.
There's two questions here. One, and then Moises Selman following.
Hi, Matt Sleeman, MedImmune. I was just wondering, though, it's a fundamental question. Do we have enough patients in the cohorts to make robust decisions? Because with this translational work, this is sort of work that's being done in the oncology field, and Joe's done work in severe asthma, where the population sizes are much bigger. And I think there's a real challenge in such a rare indication to be able to get enough patients into these cohorts to make robust decisions.
Yeah, I'd like to come on to that. I mean, a lot of what people are saying is what we should do, and ideally what would be great if we could do. But one of the supplementary questions I had to the audience, and follows on from that, is, although we know what we should be doing, is it a practical reality? I mean, certainly blood is possible. We can generate cohorts of. I mean, we're recruiting very rapidly, and getting blood is not a problem. But biopsy samples and tissue samples is going to be practically difficult. You know, the biopsy rates are higher in the USA, but still it's not 100%, and it's never going to be possible to biopsy repeatedly. So if you want to follow a biomarker over time, that's not, well, maybe in America you could do it multiple times, but you're taking a bit of a chance. No. So what do we think? Is this the practical reality? Is there something better? I mean, blood is obviously the answer, but it may not be informative enough.
Okay, there's loads of questions. Let's start with Moises, and then there's another 4 down the back.
Yes. I think that this is a critical issue also. And what we don't know is when we found a overexpressed gene or protein, if it is good or bad. I think that during the development of IPF, there is the overexpression of profibrotic but also antifibrotic molecules. So when we found something high or down, we are not sure the real role of it is an epiphenomenon, because it is increased or down because another gene, it is increased or down. And the literature is full of papers, including our papers, few, in which we found one gene, and then we obtain the knockout mice or the transgenic mice and put bleomycin and say, oh, it is a bad gene, and we publish that, and nobody repeat that, and we don't know if what we are looking in the model or in vitro in fibroblasts really has something to do with the problem in vivo. So I think that is a critical issue that we should solve. And one of the problems that I think that is going to be discussed after is that always say that there is no a good model, but nobody is looking for a model, for a good model of IPF that we can all use.
Moises, thank you. Dean will discuss models in a few minutes. But what you raise is a very good point. Of course, if you're doing science of IPF, the glory is to discover a new molecule. But actually, in terms of patients, that's just the beginning of the beginning. And I think the follow-up has not been there, partly because we have so many candidate molecules. And let's face it, our animal models have produced a lot of candidate molecules. Okay, there are questions down the back. Yes?
Yes, I just would like to emphasize the importance of also looking at the tissue in some parameters, since I'm studying, for example, mast cells, and they are not present in the bile or sputum or blood samples, and they are tissue-dwelling cells. So some changes can't be picked up in these kind of methods. And we see actually huge changes in mast cell densities and phenotypes in IPF, for example.
Okay, thank you. Well, look, while the microphone's being handed on to someone else, Hal, have you got any comments?
I just wanted to directly respond to the question about numbers of patients and do we have enough patients? We absolutely have enough patients. The problem is they're not all in one center. And I think one of the big challenges to us is to effectively collaborate and integrate cohorts. And that is absolutely doable. It's just an issue of money and time. And I think that's something that this group should really commit to because it's critical to everything you all are talking about doing. So anyway, the answer is yes, we just need to be able to integrate. Yeah.
I mean, the other thing that you need, apart from money and time, is a kind of standardized way of doing things. So the sooner we start standardizing things, the better.
Okay, we've got loads of questions. Janet Burgess from Sydney first, and then I think David Schwartz wants to say something.
I guess I'd like to echo the comments that have just been made. We've actually got a poster where we've got 3 cohorts which have been established through collaborations in this room, and we've been able to chase a molecule that we're interested in both tissue and serum from the 3 different cohorts, and I think that's the strength of this sort of research and where we need to go to take things forward.
Okay, I think we'd all agree with that. Anyone else want to comment on that specific thing? Okay, David?
I just wanted to echo Hal's comment, but also give a suggestion. I do think that part of the problem is that we haven't really harnessed the power of the individual cohorts that we have. And part of the issue is that we've developed parallel structures to deal with these patients. We have our clinical structure and we have a research structure. And we keep saying we don't have enough money for our research structure to create consistency in terms of the way we evaluate patients or the way we store specimens or the way we have access to specimens. But we're not making use of this incredibly rich and well-funded clinical structure that we have to evaluate patients, store samples, and have access to specimens. And I would offer as a solution to the problem being able to combine the clinical structure with the research structure in a way that would allow us to have consistency of the cohorts that we're taking care of and have access to the specimens across centers that are participating in a sort of a multi-centered approach to storing serum, storing peripheral blood cells, storing lung cells, and having access to those cells through the wealth of the clinical infrastructure that all of us have access to that is very relevant to the care of the patients with IPF. I'm not suggesting that we do things that are not consistent with the care of patients with IPF. What I'm suggesting is that we use that as a way of enhancing these specimens and the outcomes of those patients.
David, thank you for those great comments. There's more questioners or more points being made down the back. Yes, please. speak loudly.
Okay, this is Annika Larsson from Lund University, and I also had a post about prostacyclin, and that's you can use that as a biomarker for disease severity also, and for blood sample, and serum samples, that's perfect for analysis, but some of our biomarkers, you can't find that in the blood samples. So one should be aware that we are missing some of markers that could be really, well, giving us a point of how the disease is developing over time, and be careful and use also other methods to measure these biomarkers so in the samplings that we're taking. So that's just a remark.
Yeah, I think that the point is that blood is ideal. It's so much simpler, but it's much more complex. I mean, looking back to the early days when we thought collagen, procollagens and collagen markers would be the key, they may well be now as well. The trouble is that 90% of collagen turnover is occurring in tissues outside the lung, mostly bone and skin. So, you know, blood doesn't turn out to be a really good marker for those mechanisms. That's probably true of many things. And then there are, you know, many molecules, growth factors, cytokines, etc., that you just can't detect in blood. So the challenges are pretty clear. But I suppose what people are saying is measure things in blood just in case, because that is going to be the best option if at all possible. I think there were more questions from the back. Have you got a microphone? Oh, right. Go ahead. Yes.
I just want to echo the comments about collaboration across centres and states, and perhaps use Australia as a little bit of an example. As you know, Australia is very big geographically, and we really struggle to do any research really, or anything to do with IPF unless we do collaborate. And so I guess that's maybe why we've been looking at, you may have seen our poster on the Australian IPF Registry, which is an example of a national collaboration where we're trying to do things, as you mentioned, in terms of a protocolised fashion and collecting samples. So I agree. It's really important to collaborate nationally but also internationally. Thanks.
Thank you. Just hold the microphone for a second. So, I mean, we heard yesterday from Toby Meyer about a really good study supported by a pharmaceutical company. Is your Australian effort also supported by pharmaceutical companies, or?
It's an interesting story, starting with a philanthropic family, which an IPF.
Sorry,
keeps coming and going. An IPF specific registry. So that was where it started off, and that money was given to the Australian Lung Foundation, which is a not-for-profit organisation which sort of encompassed the whole of Australia, which I think is its benefit. But I have to say that we've been very well supported by some pharmaceutical companies since we've started. So it's not a pharmaceutical company project, but certainly we've had and welcome support.
Okay, and are you reaching out to other national bodies doing the same sort of things as yourself?
I think the important thing is to try and collaborate internationally. I think that's really important.
Thank you. Yeah.
Anne Miller.
Anne Miller.
UK. I was just going to support Giesely's suggestion about the standardisation of methodology.
Can you speak up, Anne? Sorry.
The standardisation of what we do with these samples, because even within the UK, the way in which different people process their samples is very variable. And then getting with the cohorts that we are going to have available to us in the next few years, being able to make sure that we're getting the same message across the board is very important. There is going to be a registry in the UK which will potentially store samples, but just listening earlier and talking and going around the posters, it became apparent that if we weren't all storing those samples and processing in the same way, we're going to have the same difficulties. So I would support you in that, Giesely.
Giesely, do you want to comment on that? I mean, different centres do different things. This is a bit of a challenge, but...
Yeah, I mean, I think it's fundamental, is, you know, when you want to look back historically at samples that you've collected and you then combine cohorts, it's impossible if they're all processed differently. I mean, and I think so early standardization will have really huge benefits 10, 20 years down the line.
Okay, Professor Goldie?
The studies are very expensive and supported
very nicely by pharma, because I don't think any of our funding agencies are going to have enough resources to put in against one question like this. But every one of those studies, we should be assured that they're going to be accessible for interrogation by a variety of people. The best outcome of some of the previous ways of looking at tissue has been that the information's been posted, it's interrogatable by a variety of people, and I think that, you know, like your study in the Genzyme thing, that's an outstanding way of looking at it, but a lot of different people would probably come up with different interpretations. And until we get access, that is the general interrogation for that data, you may well be taking it down one pathway when some of us might take it down another pathway. So I really think that the sooner it becomes public and interrogatable, the better it's going to be for us to get the information and make some maybe rational and collective decisions on what kind of biomarkers we have.
Okay, thanks, Jack. Other? Can I wonder, no one's mentioned imaging so far, and the importance of imaging in fibrosis. It's just been pointed out to me that Dean Sheppard has already mentioned that. But then again, well, Dean, do you want to say a little bit? Okay, you refuse to mention it again. Okay. All right, good. Gisli, any final?
Yeah.
Hal, sorry.
Well, no, so
Hal,
I
then
just wanted to ask Gisli a question. I'm always, I guess I'm the one thinking about the 5-year plan, but why isn't there standardization of processing of samples and so forth? I mean, is that something that, again, would be a worthwhile project for this group to think about, or is it just
No, it's definitely a worthwhile thing to think about. I mean, everyone, sets up their own protocols. They do what they think is best. They have their own standard operating procedures, but it's not until you have communication amongst large groups that it's the details. It's like, ingredient X in Coke. I mean, it's the bit that people always forget to say, which is actually fundamentally important. Until you have those conversations and go through the protocols, you're doing things differently. I mean, I've got a bunch of questions about harvesting cells from tissues, which we haven't got time really to go into, but that's a classic example where people do things so differently that the results from one set of cell experiments are virtually completely different and uninterpretable in the context of a different set of experiments. But so getting the SOPs right at the beginning, and there's a lot of work going on this in the UK and COPD to just get every group in the UK doing stuff the same way, and that would be, the power of that in IPF would be huge if we did that internationally. If we all said we are going to do this, this, and this at every aspect, and I think with the clinical trials it becomes easier. But then each pharma-sponsored clinical trial is going to have its own slightly different protocol, which then makes their cohort difficult to match entirely with the previous one.
Okay,
So
great.
Jeff, if I can just add one, I'm sorry, quick addition to that.
I've got a few more people, so quick.
Hal, can I just answer your direct question? Yeah. Ivan Rosas from Boston. Yeah. So I actually, I think a year ago or two, you invited me to present biomarkers of IPF, and I actually reviewed the cancer literature. And they have a couple of statements that they've made in forms of publications in which they provide guidelines. It's very difficult to tell people what to do, which I think is the point you're making. But at least they provide a written guideline on how you can actually collect samples, how you can interpret data. And then the FDA has some regulations on what biomarkers should be. So I think you're trying to say that we should maybe have a working group and accessible at least to individuals if they want to read it, if they want low-hanging fruit, and should be done.
Yeah, we've got a bit of a problem with that microphone. Richard Marshall.
Yeah, I'm just going to add the same thing, I guess. There's two sides to this. One is the clinical outcomes that we're measuring, and unfortunately we still don't have, as you've been saying, consistency across some of the very fundamental things we do. And I think we should also look at where the asthma field, COPD field, a little closer to home and respiratory, where they've got to. It's not all good news, I have to say. I've spent a number of years talking to people about sputum, And you could not believe how many different ways there are to collect sputum, and how many different, do you pick the bits out of the sputum? I'm sorry it's close to lunch, but and there's some very sort of strange but fundamental things, as you say, just mean you cannot compare like with like. But there is some low-hanging fruit. Blood samples, that's easy. We really should be able to do blood samples. Standardizing bowel, I think good luck. We've been 20 years trying to figure out how to do that. And then the other thing is actually some of the even more basic stuff, in the asthma world, PC20, it's amazing how many different ways people measure metacholine responsiveness. That just needs to be nailed. And there are some things in the IPF arena as well in terms of just basic physiology, etc. So I think there are some low-hanging fruit. But let's go for the quicker wins.
Okay, Janet Burgess, while the microphone's going to her, I'd like Gisli's comment on, if you're a young researcher and you need some resources, you need some fibroblasts, you need some lavage fluid, and you don't know quite where to get that from, what do you do? You've got a registry, there's many in Europe, they're in the States, but it's not always easy to get the resources. What would you do if you were starting out?
What I would do, and what we tend to do, is you find out who's got them and get them from the people who have them. That's what you do. You don't try and grow your own from the very beginning. You can do, but it takes a little while. But there's a bunch of groups who've got a bank of cells, fibroblasts. We have in Nottingham, UCL have them. I'm sure the Brompton have them, and everyone in the States, anyone who's got a research group and is
So you ask. Okay.
Ask, exactly.
Janet.
In terms of standardising protocols, do we as a group need to be tapping into resources that are available through the ATS or the ERS and putting out a positional paper? Is it one of the projects that needs to be put in through one of the assemblies and put out something to try and standardise things both on the clinical and the basic science side of things?
I want to wrap up on that point. I'll let Gisli have a word. Right. Do you want to very, very quickly? Yeah.
Yeah, Donna Davis from Southampton. I think there is a great example of pan-European collection of phenotypes in severe asthma, and the work that's been done in New Biopred is exactly the sort of work that needs to be done in IPF. Everybody has harmonised protocols. The potential to do this kind of work is there, but it costs millions and millions and millions of euros to do it. But I think the evidence is that Europeans can work together and they work with SARP. So in severe asthma it can happen, in IPF it should happen too.
Okay, thank you, Donna. It's a valuable comment. Can I just ask Gisli to wrap up, and if Hal has any more points, then we'll finish and then go on to the next session.
Yeah, okay, so I think, it's been a very informative discussion, and I think what's great is in the room most people agree. So we all agree that we need to collect tissue, we need to examine the tissue for stuff, and it will be most informative when we have a therapy which can actually modulate that and demonstrate the importance of the molecules that are important. But the key limiting factor is access to tissue. Lots of people have it, but we need to work together to be able to share it and to get the most out of it. And I think certainly that's coming through loud and clear, that there is a will within this group, within the IPF field generally, to work together, share tissues, and move the field forward. And I think if we get nothing else out of this meeting than some statement, some way of working forward together, then we've achieved a huge amount, actually. So I'll stop there.
Hal, do you have any final comments? No, thank you. Okay. No. Right, I'm going to ask Dean Sheppard to come up. While he's making his way to the microphone, I think, when this meeting started, 1980, it's a very long time ago, 30-odd years ago, there was a real dichotomy between the scientists, the animal model people, the human studies, and they kind of didn't talk to each other very much. I actually think it's come together much more, partly driven by the need to publish in good papers. You need to get information from all angles. But anyway, Dean is going to confront that issue for us.
So for the animal model section, we're going to have two questions. The first is one that I thought we wouldn't have to ask, but there is still debate about it, which is, are animal models useful for the development of new drugs for IPF? And I have a ulterior motive for asking that question. I'm asking it of the people who use animal models. At the ATS meeting this year, Talmadge King and I will be debating this question, so I'm looking for ammunition from the audience for my discussion with Talmadge. He's not even here anymore, so we can do things he won't know about for the ATS meeting. And then the second question is, assuming the answer to the first question is yes, what's the best animal model and what are the most relevant endpoints to measure in animal models? So let's start with the first question. Are animal models useful for evaluating potential new drugs for IPF? And I'm going to take the first 5 questions from poster presenters. So if you're not a poster presenter, don't pretend you were. Just let's give some time to the poster presenters.
Hi, Purnima Mahavadi from Gießen, Germany. For the answer to the first question, do we have good animal models for lung fibrosis or that mimic IPF? We are working with 2 animal models at the moment, the amiodarone-induced lung fibrosis model, which probably more reflects the patients that are treated with amiodarone and not much with the IPF patients in general. But we have a second model, which is a genetic model, that is Hermansky-Pudlak syndrome model, which the patients of HPS1 defects more mimic or reflect the fate of the IPF patients. So, because it's a spontaneous model and genetically altered, that could be used as a more appropriate model because it's less inflammation and epithelial-based disease. How far the HPS is involved is something that we still need to go further, but probably it's better than the bleomycin model at least, which is more inflammatory. Thank you.
Professor Maltini.
Yes, our experience has been with different animal models, such as starting 40 years ago with exposure to chronic hypoxia. Then we switched to radiation, administration of monocrotaline that also can be used in clinical work because as bush tea is being widely used in the American Indians and in southern states such as Arizona. And then eventually we have a new model of a fat embolus syndrome and by injection of triolein mimicking fractures of the bone and damage to the lungs. In all those models, angiotensin converting enzyme and angiotensin II plays a very relevant role. We found that in llamas, and Dr. Stegmark later on confirmed in Tibet with the yak that the lack of angiotensin II is ending up in no development of pulmonary fibrosis and hypertension. In radiation, we had clinical trials, especially at the Medical College of Wisconsin, where irradiation of total bodies and bone marrow syndrome for children with leukemia and severe radiation, pulmonary fibrosis and renal fibrosis have been mitigated and significantly protected by angiotensin converting enzyme inhibitors or angiotensin type 2 receptor blockers. Something similar has been done
Sorry, can I interrupt? You have made such a fantastic contribution to animal models, but I want you to finish quickly and then we can widen it to the questions asked.
Yes, just so. Yes, ACE inhibitors have been quite useful, and very recently there is a study with 47 patients with IPF where losartan has been used, and the new clinical trial will be run from several centers in the next month with application to the NIH. Thank you.
So thanks a lot, but Sue, I haven't heard any answers that would convince Talmadge yet. So I'm looking for arguments for why the animal models might be useful to predict drugs that will be effective for treating IPF.
So I would like to make a commentary with regard to, which relates to HAL original question of heterogeneity. I think we shall say goodbye to the concept that we have one model that unifies all. I think we are also on the track of recognizing the heterogeneity of IPF in terms of establishing different models. We're coming closer and closer to the conceptual view that the type 2 cell injury is probably the decisive step in the evolution of IPF, but yet we recognize there are different ways that seem to cause type 2 cell stress, DNA damage, ER stress, lysosomal stress, and probably with regard to identification of novel targets and novel therapies, we probably need to apply different models depending on what you're looking at. And likewise, I mean, still, if we find the optimum model that mimics fully human IPF, for example, the SBC mutation models in mice, still we are probably facing the problem that mice may react differently with regard to their pro-fibrotic signaling machinery as compared to humans. So I guess at the end, we end up with more models than we ever had, but we're nevertheless probably coming closer to that original thought of epithelial injury.
Thank you. I mean, what you're saying is more models, not less, but understand the limitations. Cecilia Perel, then David Schwartz, and then there's David Warburton here.
I mean, I think it's incredibly important. Animal models are incredibly important because it's the only way we can actually test the role of specific molecules. I mean, when you look at complex signaling pathways to really evaluate the role of a particular molecule, we need to utilize animal models. I mean, the endpoints then become incredibly important, and I think it's incredibly important to make sure our observations are then validated in patient samples.
Thank you. David?
IPF is a gene by environment disease, and the only way to look at gene by environment is to be able to knock out or overexpress a gene and to expose the animal to an environmental stimulus.
Okay. Bethany Moore? Oh, okay, you go ahead, and could you get the microphone to Bethany? Yeah, go ahead.
Thank you. Christina Riddell, Lund University. I just want to emphasize that there are several different models of bleomycin-induced pulmonary fibrosis. There is the usual one with the endotracheal administration, but there is also the option of give it subcutaneously via injections or using an osmotic mini pump. And we recently described that when you use the subcutaneous administration, you can actually get a less inflammatory response and a fibrosis developing in parallel with the inflammation. So the bleomycin model, when you give it intratracheally, is usually quite inflammatory, but not always. And that means everybody needs to remember that when using the bleomycin model.
Thank you.
Yeah, Jeff, I would still say that I think our animal models are the best that we have, but I think what is really important, and this is a point Martin Kolb made many years ago, is that when we test our therapies, we can't test them at day 0. Because if we just block inflammation, then we're just blocking the development of the fibrosis in the mice, and that's not really testing the therapy. So I think it's wonderful to do that as a test, but we all have to really commit to going ahead and giving bleo, subcutaneous, FITSI, whatever it is, but letting the fibrosis establish in the mice and then seeing if we can remodel it with whatever therapy we're testing. And then just as a question for the group, we've been toying at Michigan with the idea of maybe creating humanized mice and then testing some of the models that we already have in a humanized mouse system. And I'm just wondering if anyone's tried that because I haven't seen that anywhere.
Okay. David? Oh.
Here. Yep.
Okay.
Steve
all
Mattaz.
the microphones are at the back, David, so you wait.
Steve Mattaz. Oh.
Okay, Steve Mattaz.
Steve Mattaz, Western Australia. I'll get out there eventually. I think the key thing, though, with regard to animal models is that no model is going to be accurate with regard to IPF in particular, and we can't really assume that. But it's the only model system that we can use to test a particular change. So if we're looking at a particular molecule of interest, we can manipulate that. And I think the different model systems that will highlight whatever particular important molecule you have of interest is useful for testing, for looking at regulation of collagen, etc. But I think ultimately we don't have a good model for IPF. And I mean, as Cecilia mentioned, you have to test it in human samples before you're going to actually have any real human relevance to the disease.
Okay, Jack.
So I'd give you the issue that if you have a model that has got a pathway that you recognize in that model, then it's an excellent use of an animal model to test a particular drug and its activity and biodistribution. So the problem is that every one of the models has been extended well beyond what it's capable of demonstrating. And the bleomycin model, when it was first described, was a relatively innocuous model. It did not kill the animals. And yet now we've got evidence of numbers of studies and where the amount of bleomycin that's administered ends up being toxic and the animals are dead within 18 or 19 days. So that's an acute lung injury model and then maybe a little bit of response. It's not necessarily a fibrosis model. So I think that we've got some good ones. We've got an immune-mediated fibrosis. If in fact that's what the drug that you're interested in targeting is supposed to mediate, then you have the FITSI model where there's probably immunity associated with that. I think we've got a number of models, but they've been used in the wrong way for a number of studies. But if they're taken correctly, they're the perfect vehicle for discovering drugs that interfere with a particular pathway.
Okay, I think we're giving Dean a lot of ammunition for his debate with Talmage, but we're not giving Talmage much ammunition. I'd like to hear from people that are very sceptical about the animal models and what they may be doing. David Warbert.
Well, I'm sceptical about everything, but I do think animals have one big advantage, and I think that you can tile things out in the animal model and tile things out developmentally and look at windows of opportunity and stuff like this. And as I said in my talk, by the time the patients show up in the respiratory physician's office, they've already had this disease for a very long time. And if you're going to fix this disease and wipe it out, it's not treating the people with established disease that's going to do that, because it's too late. The lung is too buggered up, basically. You've got to start early. Now, I don't know how you do that in humans, because you'd probably have to irradiate everybody to look at their lungs in great detail early in life. But if we could come up with a better imaging system to do that, that's based on magnetism or something like that, that would be very, very useful. And then you could look at more early interventions to prevent progression of the disease. And of course, in the mice or rats or what have you, you can actually do that. And the other part of it is you can actually manipulate resolution of the disease and look at the back end of the injury in the animal models. That's very hard to do in the clinic when you've got a patient who's going blue in front of you. You've got to do something, doctor. So I think this is. But on the other hand, for Talmage, mice aren't humans. I don't care if you humanize the mice; they're still not humans, and the results that you get in mice may not pan out in humans. So the bottom line is you've got to look in humans and see if it's true.
So I want to make a couple of comments, and then we'll go on to, I guess, Daryl, been waiting for a long time. But so notice I didn't ask, is there a good animal model of IPF? Because that would be ridiculous. Of course, how could you have an animal model of a disease where we don't even understand what the underlying mechanism is to begin with? So it's not, do we have an animal model of IPF, but how can we use animal models to learn things that are informative about the potential treatment of IPF? So I think we've had some good discussion about that, and then maybe we can continue it for a little bit, and then we'll also come to what are the right endpoints to utilize in animal models to have them be the most informative. So Daryl.
Daryl Knight here from Vancouver. Dino, you actually stole a lot of what I was about to say. I think it's hard to model something you don't really understand. But I think perhaps sort of go to take stock from the mouse models of asthma, for example, where we've gone away from ovalbumin into more supposedly relevant stimuli, so house dust mite. But they're also now being used to, let's say, agree with endotypes of asthma. So we tried IL-5, anti-IL-5 therapy, worked beautifully in mouse, the first study, so it didn't work in humans, but now it looks like it works in a subset of humans. So it could be that the models that we are trying to use will be useful for specific types of the fibrotic disease that you're looking at, and maybe there'll be an exacerbation model, I don't know, but I think that we've got to try and ask the right questions of the model before we go any further.
Yeah. Okay, Andy, and then back to Dean. Oh, sorry.
And Henna Iclaf from Helsinki, Finland. I just wanted to inform you that dogs also develop IPF. And it might be a suitable model for the human disease, at least in some aspects. The diseased dogs are middle-aged or older. They suffer from cough and exercise intolerance. And the disease is spontaneous occurring. So it's not an induced model of inflammatory fibrosis, but it's a spontaneous chronic and progressive disease. And we are currently working on with the similarities and dissimilarities with the human disease.
Okay, I fully agree, and the mouse model is necessary for testing drugs or study the mechanisms. And the one thing I want to raise is, when you have the when you use the knockout mouse model to get something, you need to be very carefully interpret, because knockout is extremely case, and which is not. I mean, I don't think it's true in patients. I mean, you will not. You will not, lost the gene 100%. So, just my thought.
And also, all you could ever learn is prophylaxis from a knockout model, right? You can't learn about potential therapeutic intervention. So Zia's been waiting a long time.
Yeah, I've been very patient over here.
Yeah.
I think that one of the things that's missing from all the models that we have, and I guess it overlaps with your question 1 and 2, is the chronicity issue. Because everything that we do, whether the animals die straight away or recover, is a single hit. So I'd like to point out, even though he's not saying it, Tim Blackwell's model of repeated bleomycin injury, or repeated anything. I'm not saying necessarily bleomycin, but the concept of a longer follow-up, because IPF is a disease that, as we heard yesterday, probably develops over a 20 to 30-year time period, and certainly we're not doing that in the animals. And then in terms of bleomycin or other models, I think that one of the areas that we've not focused on, other than I think Landon King's group, is the resolution phase of bleomycin. Why does that actually get better? And what is there about that injury? Is it just because it's a single hit for the most part? Or is there something that we're missing about what's not actually messed up in that model that is different in IPF? And I think that's something that we should actually think about before we throw away everything in that regard. But to me, it strikes me as a very, very important feature that it actually gets better. And we haven't done much to try to mine that and see why that is.
Okay, we go to Andy. Andy, but just before you start, I'd love to hear from pharma people whether any of them have moved away from animal models, or whether that still is essential for their programs. Any people that wants to question animal models would be useful, I think.
And then Andy and Gisli has been waiting patiently too for a while. So. Not on. Not on.
It's not working, that one.
Talk about the bleomycin model, both in terms of Tim's model of repetitive injury that Zia mentioned, and also Marvin's comment that acute exacerbations and patients with progressive IPF may not be that different, in that you find areas of diffuse alveolar damage if you look carefully throughout the lung in patients with non-exacerbating IPF. And that in that context, we think there's still actually new areas of injury going on in IPF at different points of time. It's not like the IPF started 20 years ago and all the lung is being injured simultaneously. So in that context, I think, although I agree wholeheartedly with what Bethany said and need to delay treatments until after establishment of resolution,
shake it.
You still have ongoing new sites of injury, argues that still, even in the knockout situation or in the preventive situation, you're still gaining informative information about those new areas of injury. And then to speak to the problem of inflammation in bleomycin, people say, again, you have this early inflammatory phase and we don't want to just treat that. I think that's true. You do get clearly an inflammatory response, but I think many of the mouse models, starting with Dean's beta-6 knockout, have shown that inflammation in the bleo model and fibrosis are dissociable, and that, for instance, in the beta-6 knockout, you have increased inflammation from lack of TGF-beta signaling, but no fibrosis. So even though you have this early inflammatory phase, I don't necessarily think in the model it's causative of the fibrosis. So I don't think that's a lethal flaw of bleomycin. And even if we look early, I think both parts are important. What Bethany said, looking for things that can promote resolution, but also things that can still prevent development of fibrosis after injury and uncouple injury from fibrosis.
Yeah, so I mean, I agree with a lot of what's been said about the problem with the models, and you use the model to look at process rather than disease, et cetera, but I'd like to flip the question back, if you like. I mean, the reason we use animal models is that they're practical and you can answer questions quickly and easily. And so if we're not going to use an animal model to test a therapy, and given that every one of the people in this room has a different molecule and a different target and a different pathway that they're interested in, how are we going to screen all these molecules? We're going to do a clinical trial on every molecule that everyone's interested in at a cost of millions of pounds, at thousands of patients and hundreds of years.
Comment? What do you think?
Well, that would be a good question for the pharma people. I mean, it's a big challenge. How do they, basically that question is if you're in pharma, how do you prioritize your potential drugs? And they have pretty sophisticated ways of doing that, in fact. Okay, Richard's saying they don't. It's just by the seat of your pants. Do you want to comment on that, Richard, or anyone else? There's two people there. Have you got a microphone, Richard?
Well, can I just quickly comment on the longitudinal follow-up? That's possible in dogs. We follow them every 3 months until their death, and then we have access to their lung tissue because we can set the date of euthanasia and be ready to biopsy or take the whole lung. The problem in the dog model is that they are privately owned pet dogs, so we don't have a family of laboratory West Highland White Terriers, so the cohort will be small, but anyway, we can follow the disease until their death.
Well, thank you for that. Okay, let's two comments from pharma there.
Right. So,
and then we'll come back to Telmo.
On behalf of pharma, I'm not going to tell you exactly how we select our targets because it's ugly and it would take a long time. But I agree completely, I think, with what several people said. I think Jack put it best about the use of an animal model. If you are trying to target is present in a particular animal model, that's a useful whether your particular intervention hits that target in the model in which it's relevant. Of course, tell you what's going to happen in vivo in humans. At least for my part, we are trying to reconcile that with looking at tissue in humans to see, where are the targets expressed? Are they expressed in a place where we think it's likely that it's contributing to the ongoing process of disease? But it's nothing different from what any well-trained scientist would be thinking about.
Yeah, just to comment on that. I mean, I've had discussions with Ron Dubois over the years about how useless animal models might be, and I think he's had a pretty sensible view on the importance of human studies to complement. I think if we've got any target that we're going for in animal models, unless we've showed that molecule is present in human disease, it's not worth starting the animal study. I think we should all start with the humans and just analyze the presence, at least, of that molecule, and then to get at mechanisms and potential role, the animal models can play a role. But Richard Marshall.
So we have, let's do Richard and then Talmadge, and then I have to say something at the end.
So Dean,
that'll be the order of the remaining points.
So I think all the points have actually been made. So Dean, I'm going to give you the winning argument. If the question is, are animal models important or necessary for the development of new drugs for IPF, the answer is yes. For one very simple reason, is that before we dose a patient with a drug, we have to do our best to establish a dose rationale and a therapeutic index. And for that reason alone, we need an efficacy model and we need a safety model. So the answer is we are absolutely bound to use animal models. Now, that doesn't say you can't go into man without efficacy data in an animal model, but it is a much, much harder place to go and it's much more risky. But I think everybody has made the comments about using the animal models in the context of what you understand about them. we should not be developing a new model for IPF. It's futile. Do not try to do it. But I think that there are ways that you can optimize the models we use to answer the questions. One is clearly about having different injuries that generate different downstream mechanistic outcomes. So you've got a plethora of different outcomes that you can test compounds in. That's great. The other thing is you can do, maybe simple things like how you measure change. And I think there's a poster here from UCL about use of micro-CT. It was a very simple question for us, because if you look at bleo, at best, you get a doubling of collagen. You can't do a dose response to a compound when you've only got a 100%, twofold change in the outcome. So there are ways you can tweak them, but just with the questions you want to answer. Target validation is about critical mass of data coming from multiple places.
Comment?
So I wanted to add, I think Jeff said a number of things I wanted to say, but there's one point I wanted to make, and that is Joe Lasky and I were talking, and Joe reminds me that one of the things with the animal models that we have not done is allow them to age. And I think it's because of the cost. We use younger animals, and to have an animal sit there until they're a mice until he's 70 years old is very costly. So some of it has to do with us agreeing when we review grants from people who make the suggestion to not kill the grant, because the cost is a little bit higher to use aged animals. But I think if we did that, that will also help us, because we know aging is one of the factors.
So we're running out of time, so I'll just end by suggesting that I would echo what Talmud says about killing grants. So even though all of us have our own biases about one way to approach this disease, we should all admit that what we need is more creativity and a lot more information. And I would encourage people to have an open mind about any animal model that people are interested in, that you could learn something informative about. If folks can make a decent argument for why they're going to effectively learn something about a target, we should let 1,000 flowers bloom on animal models. Move on to the next topic.
Dean, thank you, and they were great message, I think, to finish with, because we mustn't let animal models polarize us in the way we go forward. Right, we're going to move to Oliver, and he'll talk about the posters on signaling and cell biology.
Okay, basically, I'd like to keep the discussion flowing in the direction that it's been heading. Just adding 2 additional points. One of which is continuing on what we've talked about, actually what GC started on biosampling of IPF lungs for translational studies and also animal models, is what are we really looking at? So most of the posters at every ICLF meeting, at every ATS, is looking at fibroblasts from controlled IPF patients, epithelial cells from controlled IPF patients, and then looking at their favorite signaling system, if you want, which we've touched upon already. And so there's 2 points I really want to like to kick off the discussion with. First of all, who's telling us that we're looking at the same cells, same origin? We all know there's an SMA-positive myofibroblast in IPF that just is not there in any other disease mostly. But we always compare IPF fibroblasts to control fibroblasts. So is this the same cell or not? Same is true for epithelial cells. They look different in disease and control, yet we're comparing both. So I want to have all your thoughts on how do we better control that? How do we better utilize what we have? Combine it with patient biopsies, maybe in different ways or means. And then the second part, I want to touch on pathways. And I think the key point here will be, yes, we all have our favorite molecules, but also at this meeting again, there are a couple of molecules that have been reconfirmed by other groups. There is always a single molecule that's floating around for a couple of years, but I think there are some themes that are re-emerging. LOXs, for example, are one, lysophosphatidic acid. I mean, there's a couple of molecules that come up from different groups, and I think that's at least the first window that we can select them. So I want to start out with the cell-specific theme, fibroblast. Who's comfortable that you're isolating the same actually origin of cell with a fibroblast from IPF and a normal lung? I'm not, but I'd like to hear your opinion.
Okay, particularly from people, can we start with the poster presenters? I don't think, Stephen, do you call yourself a poster presenter? Yes. All right, okay.
Let's have a vote on that.
Look, I think it's a very important area, and some of the cells that we've collected, for example, if you look at most profiles when they compare normal fibroblasts and IPF fibroblasts, there's always a large overlap at the bottom, and you've got a few outliers at the top. It's just consistent. What we actually found when we actually forgot about comparing IPF and normal and put them all together and just said these are an arrangement of fibroblasts, how do they respond to different things, they all polarize into two different areas. And I think what the problem is that when we culture so-called IPF fibroblasts, we don't know what they are. I mean, Darryl, for example, has shown the heterogeneity within the lung. If you take fibroblasts for different areas of an IPF lung, they all behave very differently. And I think, for example, you're going to have some normal-like fibroblasts in the lung, and we might be isolating those. We're also isolating the cells that grow quickly, which are probably not the myofibroblast phenotype, which is what we may be, or should be looking at when we're talking to IPF. So I think we've got to be very careful when we just say they're IPF because they've come from a patient versus normal, and maybe those normal aren't exactly normal all the time either. So I think we just be very, very careful what we actually, are looking at, because I think we miss things along the way because of doing that.
So how do we do that, to be careful?
Okay.
You have to phenotype each cell line as they come out and basically do the comparisons based on phenotypic analysis.
How do you phenotype a fibroblast, the way we isolate them?
Well, and that's a very good question because, because a lot of them aren't going to express smooth muscle actin. Maybe somebody else can come up with how to do it.
I get a bit of a deja vu here. I'd like to...
The left side.
I'd like others to get involved. So, I don't know if Ganesh would like to comment, because actually, these debates have been going for a very long time, and in the 80s, Ganesh was one of the first to describe fast proliferating cells. And sometimes when we have these discussions, I wonder if we've moved on that much further, but I agree, we need very good ways of characterizing these cells. So, just to get a historical perspective, I would like Ganesh's comments.
Thanks very much, Jeff. So, Ali asked a very good question. How do you phenotype the fibroblast that comes out from the biopsy sample? And Steve also made a very good comment already. You've got to be careful, and Ali said, how are you going to be careful? What we learn from the fibroblast that comes out of that little explant, and from where that little explant comes from, is extremely important, because by the time that the cell doubles and double cycles or whatever in the culture dish, it's already been in vitro aged, if you will. So there is so much of epigenetic factors that goes on in that culture dish, and we've got to be so careful in extrapolating. So what we learn from that is a very tiny speck of what is really going on. And the reason that we are all dumbfolded in terms of the IPF heterogeneity is concerned because of the heterogeneity within cells. The cells from the honeycomb lung are senescent cells. They're already aged, so you're going to be apoptotic cells from there. The cells from the relatively normal are normal cells because they're all diploid cells. None of these is malignant cells. So the problem that we are going to be trying to extrapolate from the fibroblasts that you're going to get from the explant is really not that you're not going to get to the bottom of it. So because there are so many ways of how we are going to do the transbronchial lung biopsy. There's so many ways of how we are going to get the lung from thoracotomy approach or so, or from explant. So I think it's going to be a very, very difficult situation to extrapolate from the fibroblasts that you're going to learn from the IPF lung.
Okay, we have another.
So I'd just like to make a plea that we try not to be too dogmatic about this stuff. I think we can learn a lot from cells that we get from explants, whether they're epithelial cells or whether they're fibroblasts, but there is a kind of pervading dogma, if you like, that's only certain cells are relevant. So, for example, in epithelial cells, 82 cells are the cells, and if you don't have 82 cells from patients with IPF, then your experiments are rubbish. I just don't think that's true, because we know we know nothing about the cells from epithelial cells from IPF. As Marvin said yesterday, the small airway epithelial cells are likely to be important. The work on MUC5 would suggest that maybe bronchial epithelial cells are important. We don't know, so let's get the information out there, make sure the experiments are done appropriately and the protocols where we can are standardized and realistic, but let's not just say, well, this can't be true because it doesn't fit with what I believe to be true from 20 years ago.
Okay. Okay, sorry.
This was just in a quick response to phenotyping fibroblasts. There's a professor in Stanford who's looked at homeobox genes from different fibroblasts in different regions of the body, and you can see different developmental fates and actually see that the genetic profile of these homeobox genes is very different. And I think that would be quite interesting to explore whether the fibroblasts in IPF are resident or whether they've actually come from fibrocytes or a different cell type in the periphery. And I was wondering whether you could look at their homeobox profiles to elucidate the fate of where these cells are coming from.
Okay, I mean, it'd be great if we could get some discussion about fibrocytes, actually. I don't know if you'd be happy with that, Oliver, but, there's quite a debate on their role. So if you feel that fits in, we could go on to that. But Janet Burgess.
I think we need to be careful being hung up on alpha smooth muscle actin as the marker. It isn't a distinguishing marker necessarily for a fibrocyte or a fibroblast, or the smooth muscle cells also express alpha smooth muscle actin. There's a paper by Singh comparing fibroblasts and smooth muscle cells from the lung, and there's basically no difference between them. There's no way of distinguishing these cells. So I think I'd like to echo Gisli's comments that we need to just make sure the experimental protocols are optimised and then interpret the data as it comes out, rather than getting hung up on what type of cell people are using.
So, I mean, the state of the art in this field now is single cell analysis. And each cell is potentially different. And so the question is then, how do you deal with that complexity, and how do you analyze that complexity of information when all the cells are expressing 40,000 genes in a different pattern, and potentially all these genes can be manipulated depending on which transcription factors are dancing on the head of which pin. So I think in the lung, I mean, we know there's at least 2 sets of fibroblasts in the lung, and there's probably many, many more of those, referring to the comment about the homeobox story. And so I think within IPF lesions, there's going to be dozens of different phenotypes of cells, and it all needs to be analyzed cell by cell, and that's just how the genome actually does it.
Okay, I think Euben's got a microphone here. Euben Moodley.
Euben Moodley, Australia. I think the field in terms of resident cells has moved on relative to fibroblasts, because we now talk about mesenchymal stem cells that are resident within the lung, and that they look exactly like fibroblasts. And from my perspective, I don't know the difference between a mesenchymal stem cell and a fibroblast. So there may be, in essence, a mesenchymal umbrella of cells within the lung, and each of them have subpopulations that respond to different stimuli injury, and they may actually then also produce fibrosis or other responses. And I think it's important in the context of lung fibrosis to, I think, reevaluate the hierarchy of mesenchymal cells within the lung relative to health and disease.
Thank you for that.
I would like to make a brief comment about fibrocytes.
Moises Selman.
Moises Selman. There is a particular problem with fibrocytes because they lose their characteristic markers when they are in tissues. So perhaps a few days after they arrive there, they lose their markers. It is very difficult to obtain a good picture with these cells in vivo in the lungs. So it is an additional problem.
I mean, this issue of plasticity and the rapidity with which cells lose markers is very important. Oliver.
David. So, as a non-cell biologist, I'll make a comment about cells and pulmonary fibrosis. I would just suggest that we don't have enough work in the area of this complex milieu that the cells are acting abnormally in. And we need to invest in systems that allow us to stress cells in the way that they're stressed in the IPF lung, either through other cell-cell communication or through the very abnormal stress that those cells are under, either oxidative stress or stress of the lack of flexibility of the lung and those abnormal ways in which cells could respond in a cell dependent type of context.
Or putting them in an extracellular matrix from a diseased lung, like people are doing
or putting
now.
them in an extracellular in a lung, but also creating media that allows you to alter the ways in which cells are stressed and cells respond to that stress, either by creating different types of matrix that has more or less tension in the matrix, and to understand how the cells respond to that.
Okay, we've got loads of hands going up. So Martin's got a microphone, then we'll go to Andy Tager.
Okay, Martin Cole from McMaster. I had to jump up because you said fibrocytes. So the first paper that I read that brought me actually to fibrosis was called Anchorage Independent Growth of Fibroblasts, and there was a debate going on between Ganesh and Jack Goley in the mid-'80s. I was a clinical doctor, I had no bloody clue what that means, but it basically says what you just started to allude to is that a cell in a wobbly matrix is quite different from a cell on a stiff matrix, which is plastic dish. So when Ollie started to ask, what fibroblasts should we use to compare to what, it depends widely on the question you ask. For a lot of questions, that's a wonderful system. For others, it might be totally useless, similar to the animal models. When you think that our cell that we look is a fibroblastic foci, myofibroblast, and we know these things don't proliferate very much, they make a lot of stuff. And when you just mince your tissue, you look at cells that grow out of that tissue. Those are not the cells that sit in the fibroblastic foci. Yeah? So you still think that these cells reflect what happens in this focus, which is probably totally wrong. But it's still interesting and important to study that stuff. When you look at the fibrocytes, it's the same thing. When you look at a fibrocyte as a cell, you're lost, because this cell is not a cell. It's something that goes from A, bone marrow, to B, which is lung or kidney or liver, and it changes its phenotype and it changes its hair color like a young teenager nowadays. So insofar you can't really look dogmatically at these things. You have to look at where do you collect them and where do you grow them, expand them, and how you interpret what they do there. A fibrocyte is never made to live on a dish. So when you get a cell out of a blood sample and say this is a fibrocyte, does X or Y on a dish, that's not a fibrocyte how it's in our body, and it's not a fibrocyte how it's in my lungs, as opposed to the lungs of our IPF patients. Some of them sit in the audience. This is totally different, and that's where you come back with that matrix.
Okay, just while you've got the microphone, do we know that they go to the lung and make matrix and are contributors to fibrosis?
Well, we know that they go to the lung. I don't know if they make matrix, but they go to the lung. They show up, and Moise showed that nicely. They show up and they're close to fibroblastic foci. If they make the focus, the cells, I don't know. They might influence it.
Yeah, okay. Andy, have you still got a microphone?
Yeah.
Okay.
So coming back to Oliver's initial charge of trying to compare fibroblasts or any cell from IPF versus normal, so I think an alternative way to phenotype the cells or characterize the cells, in addition to how fast they proliferate and how much they migrate and what cytokines they make, comes back to what David was saying, that this is a disease of gene by environment. And we think the mechanism that environmental stresses, complex as they are, translated to the genome is by epigenetic modification. So could we basically phenotype or categorize the cells ultimately by their epigenetic modification, comparing epigenetic marks in the fibroblasts or whatever cell from the IPF lung, which should be stable, to the epigenetic marks that may or may not be present in
Well, I know
normal.
we had a poster on epigenetics of fibroblasts from patients with IPF presented by Robin McAnulty, and I think Toby Mayer was on that as well. Do they wish to comment on where they are thus far on those studies? I think they're in the other room. All right, well, if they heard me, then maybe they can make their way to a microphone. Yes.
So I just wanted to say something similar to Andy's comment just a minute ago. So in terms of the way this discussion has progressed, we just, before this, discussed whether we should be studying animal models. And I think you could also ask whether we should be studying primary cells, because not only do they have the issue of the heterogeneity that Oliver has been discussing, but these cells have been in an abnormal environment for a very long time. And my bias is that you're just as likely to find responses to injury and trying to, in a teleologic way, heal the ongoing injury as opposed to the primary injury event or the mechanisms of disease. And so I think a real challenge that's been difficult for us and others in the field is to try and understand how to pick out what may be a primary injury mechanism as opposed to a homeostatic response or a beneficial response of these cells. And it's compounded by the differences that you see from one biopsy to another or one cell line to another. And I'm interested in other comments for this, but that's one reason that I think that the animal models have some additional value, is because at least you know when the injury happened and you can do some temporal evaluations there.
Okay, great. Well, we've got loads of, I think there are more questions from the front, so let's keep the microphone going that way. Robin McAnulty, I can see who must be in the other room, is going to come back to us on epigenetics.
This is a test. Yep, so we've been looking at the methylation profiles of fibroblasts from IPF and SSC. There are studies that are at a very early stage at the moment, but there are clear differences between what we call control fibroblasts, IPF and SSC. There's a great deal of heterogeneity there, so that fits in with the rest of the disease problems that we have. And I think we've just got to sort of follow this a bit further. They're preliminary studies, and hopefully in the next meeting we'll be able to say a lot more about what's going on.
Robin, so I think the message is that there is definitely changes, but understanding the details of that and what genes they affect is still ongoing. Is that correct?
Yes.
Okay, thank you. Ganesh, have you got it? Okay, let's go on this way. Yeah.
So
Dean.
Excuse me, as we just heard, I've found this discussion extremely informative in relation to animal models. So, the basic binary argument is that IPF's a human disease, so we should be focusing on human cells and tissues in vitro before we make any decisions about what drugs to test. And I would argue that although the animal models are all imperfect, at least we understand how to isolate specific pathways very well in these models so that we can potentially study them. And I completely applaud efforts to improve ways that we could utilize human cells to try to do that. But I would argue that this discussion points out that we're very far away from being able to do that. And so if we put up a barrier and say we shouldn't be designing new drugs based on animal models, we have to use human cells and tissues, I would argue that we're all going to die before we have new therapies for IPF. And personally, I'd like to live to see some effect that actually has an impact on our patients.
Oh, that's a fair comment. Thanks, Dean. Yeah,
Jack. So it's intriguing that most of the conversation is about fibroblasts or myofibroblasts. And that's really because those are the only cells we can culture out. I mean, the practicalities are we've failed miserably to culture the epithelial cells in an adequate way, to keep them alive, to interrogate them correctly. And then I think the other thing is that we have to be careful to examine the behavior of the cells you get out as soon as possible. I'll tell you the one story where we were isolating these anchorage-independent fibroblasts from the lung. That was great. But Manel Jordana was looking at proliferation characteristics and certainly characterized fast-growing lines and slow-growing lines. So he then went ahead and cloned from those and isolated a fast-growing clone. That was great, but it had a Gaussian distribution of other proliferations in the primary line. The fast-growing clone was then recloned and gave rise to a Gaussian distribution of fast and slow-growing lines. So these are very non-permanent changes that we see. And I think if you're going to use fibrotic lines versus non-fibrotic lines, you have to interrogate their behavior, whatever it is you're looking at, within 2 to 3 to 5 passages, and no more than that, to be interpretable in order to have look at the differences that you're after. And if we can, let's get into the epithelial cell culture better, because those are going to be equally important and informative and be able to get the information out of the IPF lung.
Okay, Gisli has a point to make.
Rachel.
Sorry.
Rachel Chambers.
Rachel Chambers, sorry, and then Gisli.
I just want to sort of echo the kind of complexity that we're faced with when we do our in vitro experiments. And there's 2 points, or maybe 3, I want to make about when we isolate these cells, the isolation protocol itself dictates what phenotype the cells are going to have. And Chris Scotton, who's sitting at the back, has got a PhD student who's just using different isolation procedures, and you get a very different alpha smooth muscle positive, active negative or positive population from the same biopsy, depending on how you're doing the isolation. So that I find completely baffling, because obviously now we don't even have, know how we should isolate these cells within a single patient biopsy and know which population we want to study. The other thing that I was struck was at the ATS this year, there was a presentation by Craig Henke's group showing how important the matrix is in terms of conditioning the epigenetic, phenotype of the cell. And I think what their data showed is that the matrix dictated the similarities between the expression profiles greater than it did between patient and non-patient derived cells. So it was the matrix that was much more important. So again, we're growing these cells on tissue culture plastic. Martin's already talked about that. A fibrotic focus may resemble hard tissue, but normal lung certainly doesn't, and that's about 5 kilopascals versus 100. So we're very different isolation procedures, very different culture conditions. So where I think some of these cells might be useful is actually trying to understand the pharmacology one is interested in terms of drug development. So when we look at control cells, fibroblasts from IPF, from non-IPF, they might behave very differently. We don't know why, because we don't know what the culture conditions is doing. But what we can use these cells quite well for is to do good pharmacology and to dictate whether dose responses, whether mechanisms are actually replicated in the diseased cells, because sometimes just basing these on normal cells might be actually quite misleading. So those are the 3 things I wanted to say.
Thank you. Did you want to comment?
Yeah, no, again, I just want to echo a lot of what has been said, in that, it's like the animal model discussion. These are model systems, and as long as we recognize the limitation that every system has, the information that can be gained is valuable. So let's not, disregard stuff, but let's just acknowledge the limitations and use them as model systems.
So, Gunilla Westergren-Thorsson from Lund University. On the same line as Rachel, I think we simplify the fibroblasts too much, and we need to have more biomarkers for what is really a fibroblast. And for example, also combine culturing with looking in the tissue. For example, fibrocytes are really combined with structural changes of disease. And also mesenchymal stem cells do have another pattern than fibrocytes if you look into disease and disease state. So I would favor more combining both histology and culturing methods, and also to be aware of disease state, because those fibroblasts do really behave different in different disease state.
Thank you, Gunilla, and you've had an enormous amount of experience with cells, so they're valuable comments. David Warburton.
Yeah, so we all have this 18th or 19th century illusion that we were taught in medical school that the mesenchyme is boring and not moving about very much, and things are stuck, and we think that when it's a scar that stuff is stuck and fixed. But actually, if you look at development anyway, the mesenchyme is a hugely busy place, and cells are crossing over each other and crawling over each other and exchanging compartments all the time. We know absolutely nothing about this in scarring in the lung, and I bet you that there is actually a lot of cell motion in these scars, and that the cells are not actually stuck. So that would be a very interesting thing to know about, because that would affect the biology of the cells in terms of their signaling and their response to mechanical force and everything else.
Okay, I think we're getting towards the end. Any more burning statements from the audience? We've got what? Janet Burgess wants to say something. Have we got a microphone over there? Oh, you've got one.
I've got one.
Excellent, thanks.
I think the importance of the matrix can't be overlooked. Dan Schrimpeling's shown very nicely that the density of the matrix, if you put fibroblasts from the same origin on matrix of differing density. Their proliferative profile and their migratory profile is completely different depending on the density of the matrix that they're on. And we've shown that if we take cells from asthmatic individuals and get them to lay down matrix on tissue culture plates, remove the cells but retain the matrix, and then put cells that are derived from supposedly normal individuals on top of that matrix, you can change the way that those cells behave in terms of their proliferative profile. We've got a lot of information now that the matrix is different in IPF. There's lots of biomarkers that are coming from the matrix. So I think that understanding the matrix and how that matrix is being produced by the cells in the IPF lung is going to be central in understanding the disease process.
Yeah. Many years ago we showed matrix and mechanical forces are absolutely vital to determine how a fibroblast behaves. So thank you for that comment.
And just one more thing, that there'll be a major scientific symposium at ATS next year looking at how we can, the advances that are being made in ways that we can understand the matrix and the cell interaction with it.
I want to hand back to Oliver in a second, but Richard.
Just a very quick comment. Rather in the way we got to with some of the clinical discussions around standardization, I don't think we should be striving to standardize an in vitro protocol, but what I think would be useful is if there was some consensus about how you characterize an in vitro model. In other words, are there particular markers of the phenotype of the cell? Are there particular things that should be described in some detail about how they're isolated? I know we have methodologies, but I think if we could start to build a consistent way of characterizing the in vitro models, that might be a step forward. It's not to sort of reduce the number of ways we do things necessarily.
Well, as you've already all realized, we have a fantastic organizer for this meeting, and I know that he would like to get some sort of consensus coming out of this meeting eventually. So we may try to publish something that addresses some of those issues along those lines. Oliver.
Well, I think this has been an incredibly rich and intense discussion. I think what the poster session has actually shown us how actually deeply tied those 4 topics are. I mean, we come back to the same issue. It's not like we have 4 different communities here that have independent discussions, and that's actually very comforting. I'd like to close by borrowing a sentence, Dean, that you said. I think it's incredibly important to go with what we have, because what we have in fibrosis is superb. I mean, we as a community have probably moved the field farther than any other fibrotic disease lately. I mean, you know, liver fibrosis, they don't have that much of a good model and a drug, and kidney fibrosis doesn't. We shouldn't undersell our field. We tend to do that. So that is that is actually one conclusion. But on top of what we have is, I think I'd like to leave you with three opinions. One is we need to define better the matrix, because if we put our cells into a diseased matrix, they may completely act differently. Maybe a normal cell becomes a fibrotic cell, and we don't know how to isolate the matrix. I think we need to invest as a field into matrix isolation. Yeah, you've started 25, 30, 40, how long ago? Years ago? Long time ago, yeah, with the matrix. But still, we cannot properly isolate and define the matrix composition IPF. I think that is one field. And secondly, I think we need to really standardize defined cell culture conditions. I mean, partly epithelial cells have moved ahead because we fax sort them, so we are pretty sure of what we have. I mean, Rachel, you said it, fibroblasts, they're a different isolation methodology. They don't rely on fax sorting, so they change phenotype during isolation. So we need to invest in that. The only marker, I mean, Moises has been Taiwan, you and Jim have been working on for a lot, but of course that's lost in IPF, so we can't use it to fax sort. And I mean, Paul and Jason and Bridget, you've described that the mesenchymal compartment is multifold, yeah, NG2 positive parasites, what do you have? So I think that's an area we should invest in. And I think we need to go also into new challenges and new exposure settings, like David has mentioned. Like, you know, we need to find new environmental exposures that may be relevant for cells and animal models. For me, it's been a great session. I thank you all for the discussion. Jeff, back to you.
Well, Well, thank you, Oliver. I'd also like to thank Hal and Gisli and Dean. I thought they all led their discussion superbly. Thank you to all the poster presenters. I hope this has been vaguely useful for you. We haven't been able to focus on your individual poster, but we hope you got good feedback outside. So thank you to all the presenters of posters and all those who have asked questions. I want to wrap this session up, but hand back to the organisation. Thank you.